Good morning, students. Today we are going to discuss genetic disorders. So, today we will discuss genetic disorders in human being. Now, the question is how to know the genetic disorders and in the human beings. There are many disorders or disease occurs in the human being. The question is how to know the pattern of inheritance of these disorders in human being. We know that in Mendel used the pea plant and he crossed the pea plant to know the pattern of inheritance. So but in animal or in plant, this controlled cross to know the inheritance of traits is possible, but in human being it is impossible to do the controlled cross to know the inheritance of a particular trait due to social issue. But in the plants or in the other animals, that like the Morgan used the Drosophila for his experiment to know the eye color in Drosophila. Mendel used the pea plant for his experiment to know the inheritance of traits. Likewise, question is how to know the inheritance of traits in human being. So today especially we are going to learn the different genetic disorders that found in human being, and we try to learn that how these traits pass generation after generation, or we can say that what is the pattern of inheritance of these traits or these disease or these disorders in human being. And just now I said that in human being it is not possible to do the controlled cross between the two human being and know the inheritance. The question is then how to know the pattern of inheritance of human being. So to know the pattern of inheritance of human being, we can use this pedigree analysis. This is the tools to know the pattern of inheritance of gene or pattern of inheritance of any trait in human beings. The question is, what is this pedigree analysis? In pedigree analysis, we use the family history. We make the family tree. Suppose we want to know any particular trait, that how this trait passing in a family. So we first of all we find that the different generations of that family, that in which generation, in which person, male or female, that trait or that disease was present. And in this way we make the pedigree chart, or we make the family tree or family history, and by the analysis of that family history of different generation, that in which generation that particular disease appeared in which person, male or female, like we can know the pattern of inheritance in human being also. So to make the pedigree chart, first of all we use the different symbol to make the pedigree chart or family tree. So this square symbol we uses for the male, this circle uses for the female, and if the square or circle is colored, means it shows that that person is affected for the disease, that is affected person. And if the male and female are attached by this horizontal line, it means they are mating. Mating means they doing the reproduction. Likewise, suppose this is the parent, and if we give this vertical line, and in vertical line then we show the children in the order, that this is the first children, now this is the second children. Likewise, suppose this is married with a female, now again we move forward. So we make the chart, family tree of that family, and by the analysis of that family tree or family history, we can find out that how a trait, how that particular trait, how that particular disease passes generation after generation, means what is the pattern of inheritance we can learn. But before going to analyze any pedigree chart, some points we should keep in our mind. First of all, these genetic disorders, first of all, you know that you learn that the characters are controlled by the gene. You learned in the Mendel's inheritance also that the genes are responsible for the trait. So for genetic disorder that occurs in human being, this is also controlled by the gene. And you know that the gene present on the chromosome, we already learned in the chromosomal theory of inheritance that the gene found on the chromosomes, and also we learned that this gene is made up of the DNA. So DNA is the genetic material that carries the information generation after generation. So DNA replication occurs, and the copy of DNA passes from one generation to another generation, and it carries the information of traits from one generation to another generation. Normally there is that DNA without change passes generation after generation, but occasionally sometimes some changes, some alteration in DNA occurs, and due to the change in DNA some genetic problem or some abnormal disease, some abnormal problem develop in the human being, and this change in the DNA or gene is called the mutation. So due to this mutation many disorders occurs in the human being. Now today we are going to learn some Mendelian disorders. Actually there are two types of genetic disorders. Some are Mendelian disorders, some are chromosomal disorders. Mendelian disorder are the disorders which follow the Mendel's pattern of inheritance. That's why they are called the Mendelian disorders, because they follow the same pattern of inheritance that the Mendel studied. So Mendel said that the one gene is responsible for a trait. So in this Mendelian disorder also, if there is the change in that gene, that particular gene, there will be the disorder. So this Mendelian disorder is actually determined by the change or mutation in the single gene, because we know that the Mendel said that the one gene is responsible for a trait. So because Mendelian disorders follow the Mendel's pattern of inheritance, so definitely this type of disorders are determined by the mutation in the single genes. So this Mendelian disorder can be traced in the family tree or in the family history. We can trace this Mendelian disorder because this disorder transmitted to the offspring on the same lines as we have studied in the principles of inheritance or Mendel's principle of inheritance. This follow the same pattern. It means the pattern of inheritance of such Mendelian disorders can be traced in a family by the pedigree analysis. So many Mendelian disorders are found. For example, hemophilia, cystic fibrosis, sickle cell anemia, color blindness, phenylketonuria, thalassemia. These are the Mendelian disorders that follow the Mendel's pattern of inheritance. We know that Mendel said that the traits may be dominant, may be recessive. So these Mendelian disorders also may be dominant, may be recessive. Means these diseases that we are going to talk today, that traits or that disease or that disorders also may be the dominant or may be the recessive. Another thing is that gene may be present on the autosomes, may be present on the sex chromosomes. So these Mendelian disorders may be autosomal linked or may be sex linked. So if the disease is autosomal dominant, if there is the autosomal dominant trait, then how to understand that the trait is autosomal dominant? So one clue I am giving here: if the pedigree seldom skips a generation, it means if that trait appeared in all generation, it means that trait may be the dominant, because in the dominant autosomal dominant trait, the pedigree seldom skips a generation, means the rarely skips a generation. It means we can say that trait found in all generation. So if in the pedigree chart, if a trait is present in all generation, so that trait may be the dominant. But if the autosomal recessive trait, suppose this is the recessive trait, then the pedigree may skip a generation. Suppose in the pedigree chart, suppose a disease is not present in any one generation, so that pedigree, it means showing that pedigree is the trait of recessive condition. So to identify or to know that the trait is dominant or recessive, very easy thing we can see. If the trait is present in all generations, so that traits may be the dominant, and if the traits is not present in all generation, it means that trait may be recessive. So autosomal dominant trait seldom skip a generation, while the autosomal recessive trait may skip a generation. Now the third point, if the trait is sex-linked dominant, if the trait, so that will be the more common in females. If the disease is sex-linked dominant, so that will be more common in females. And if the sex-linked recessive trait, if the disease is sex-linked recessive, so that will be more common in males. Sex-linked disorders we have already learned, like hemophilia, color blindness, we learned that follow the criss-cross pattern of inheritance. So this sex-linked trait follow the criss-cross inheritance pattern, means the passage from male or father to daughter, and from the female a carrier mother to son. It passes. It follow the criss-cross inheritance. So if in the pedigree, if we, on the basis of this point also we can understand that the disease is autosomal or sex-linked. And if the disease is Y-linked, that it directly passes from father to son, because the father provides his Y chromosome to the son, not to the daughter. While we know that the mother provides her X chromosome to both son and daughter. So if the mother is carrier for the gene, if the mother is carrier means phenotypically normal, but she has the gene for the disease, so that mother may pass the gene to the sons, So this sex-linked disease or sex-linked traits follow the pattern of criss-cross inheritance, means male to female and female to male. Now you see a pedigree chart that is given in the NCERT book also. Suppose this is the chart. Here this is the female marry with this male, and now these are the children of, this is the first generation, parent generation. Now this is the second generation. Suppose this one is the 1, 2, 3 and 4 children. This is the first, this is the second, this is the third, and this is the fourth. This is the female, or this is the male, this is the male, this is also the female. Suppose this male marry with this female, and now this is the third generation. These are the 3 children. Likewise, this, suppose this daughter also marry with this male, and these are the 3 children of this third generation. Now the question, suppose this chart is given and the question asked to you that identify this given trait. This is the pedigree chart for a trait. So suppose given question that you identify that the given trait is dominant or recessive, एक question तो ये हो सकता है. Likewise, suppose ask that the given trait or given disease is autosomal or sex linked. So then how we can identify? So these are the points helps us to identify this disease. So now you see that this disease is present in first generation also, second generation also, third generation also. Means that trait is present in all the generation. It means this pedigree or this trait or this disease may be for the dominant trait. And because here showing the both male and female also, we know that if it is the sex linked, so either may be common in female or in the male, but here we can see that the trait is present in both male and female, so it may be the autosomal trait, means the gene for this disease is present on the autosomes. Now the question asks that now you give the genotype of each individuals in this pedigree chart. So what will be the genotype of this individual? So suppose first of all we should identify that this given chart is dominant or recessive. Then we should identify that this is the autosomal or sex linked. So because this trait is present in all three generations, so it may be the dominant trait. And also both male and female are affected. Also it is not following the criss-cross inheritance, so it may be autosomal, not sex linked. Now how to find the genotype of each individuals? So we know that this trait is dominant autosomal dominant trait. So we can use the symbol A for the allele, capital A we can use for the dominant allele, and a small a we can use for the recessive allele. So because we know that this trait is the dominant trait, this trait is the dominant trait, it means the individuals having the disease, they are the either homozygous, means either a capital A, or it may be heterozygous A. But those individuals which are not affected, it means they are recessive. It means, and we know that the recessive trait always present in homozygous recessive trait. Homozygous recessive condition means their genotype will be a small a. This will be also a small a. This will be also a small a. This will be also a small a. This is also a small a. This one is also a small a. This is also a small a. This one is also a small a. This one is also a small a. Now we left this only disease, but so this because this is the dominant trait, and dominant trait can appear in both homozygous condition as well as heterozygous condition, means their genotype will be either capital A or capital A small a. So suppose मान लिया जाए कि their genotype is capital A and now marries with this person having the homozygous recessive allele. So in that condition, there is the capital A and a small a. All individuals will have the heterozygous condition. But you know that there is the small a, this is also a small a, this one is, so then how this will produce? It means definitely this person is not homozygous dominant, this is heterozygous dominant, because if we take here capital A, small a, in that condition the both type of individuals will produce capital A small a also and a small a also, because it will produce one type of gamete, it will produce two types of gametes. So capital A when fertilizes with a small a, this is the genotype of progeny, this small way fertilized with this small, then means in that condition it is possible. So it means that individual is not homozygous dominant, that will be the heterozygous dominant, means capital A small a. In that condition only these two types of this genotype can be appeared. So this will be also heterozygous. This will be also heterozygous. Okay. Now so this is heterozygous. This is the homozygous. Now these are the offspring. So that will be also heterozygous. So we can learn all the genotype of this condition. So this is the autosomal dominant trait. For example, one disorder is myotonic dystrophy. This is the muscle problem. In this problem, that this is the autosomal dominant trait. Now we will talk in the next video.